Topic 7 Modern Genetics 7.1 Using Gene Sequencing. How much do we as humans have in common with these organisms? With chimpanzees it is approximately 98%, with zebrafish is approximately 85%, with bananas is approximately 50%, and with bacteria is approximately 7%. How do we know this? We know this because different organisms have genetic materials, and we could then look at the genetic material such as their DNA by looking at the DNA sequence, and when we align that DNA sequence we compare the similarity to produce an evolutionary phylogenetic tree, and the more closer related we are, the more similarities we'll find in the DNA sequence, which means that we have a more recent common ancestor. To do this, we need to first obtain DNA, and that requires DNA extraction. In a crime scene, there are many possible sources of DNA evidence where you could obtain the DNA, such as blood, skin, saliva, tissue under nails, hair, and semen. In topic 1, we learned how DNA is replicated during interphase of the cell cycle, and this requires the use of different enzymes. First, DNA helicase is used to unwind and unzip the two DNA strands by breaking hydrogen bonds. Then, DNA polymerase joins the three nucleotides to DNA strand, which forms hydrogen bonds between complementary base pairs. And then these nucleotides have to be joined together. So this is done by DNA ligase, which joins sections of adjacent DNA nucleotides together by forming phosphodiester bonds between the phosphate and deoxyribose sugar of adjacent DNA nucleotides. Now we could use the same idea where if we were to get DNA from a crime source, the sample of DNA is very likely to be in low quantity. So if we want to make a larger quantity of the DNA, we could replicate the DNA using similar enzymes. And this involves the use of PCR, polymerase chain reaction. And in the PCR machine, you use enzymes and you also use different temperatures. First, the DNA strand has to be separated, and this is done around 95 degrees Celsius, known as denaturing. A primer then attaches to the DNA strands, and that is then extended by DNA polymerase. This produces 2 DNA molecules if you start off with 1, but if you were to continue repeating this cycle, you could then make 4 DNA molecules, then 8, and so the more cycles you do of PCR, the more DNA molecules are produced, and therefore you are increasing the quantity of the DNA sample. Explain the term genome. A genome is all the genetic material of an organism, and in eukaryotes, the genome, the DNA is in the nucleus, the mitochondria, and if it's a plant, DNA could also be found in chloroplast. However, in prokaryotes, because they do not have a nucleus, the prokaryote genome is in the nucleoid and also plasmids. Describe how sufficient amounts of DNA can be obtained in preparation for gene sequencing. So this is what we just mentioned. Obviously, we want to extract DNA from our sample. So a DNA sample is required, and that could be extracted from cell. And then if we want a large enough quantity of that sample, we want to do DNA polymerase chain reaction. And in the exam, you'd want to mention the different temperature requirements. So 95 degrees Celsius is used to separate the DNA strand. A primer then attaches to the separated DNA strands, and that happens around 55 degrees Celsius. And then DNA polymerase extends the primer to form the daughter strands, and that happens around 72 degrees Celsius. And this whole cycle would then have to be repeated several times so you could obtain or make a large enough DNA sample to be used. So once you've amplified the DNA samples by PCR, it can be used in gene sequencing to predict different things. So we learned from protein synthesis that our genes have a DNA sequence or order of bases, and that order of bases can be copied in transcription to produce mRNA, which is a copy of the DNA genetic code, and then the codons in the mRNA codes for specific amino acids to produce the sequence of amino acids, and that uses the genetic code. So we could see that AUG codes for the specific amino acid MET, and then each codon will code for other specific amino acids, which will then produce the sequence of amino acids until you reach the stop codon. And this means that we now know the sequence of amino acids. So by amplifying DNA samples using PCR, we can then use it in gene sequencing to predict the amino acid sequence of proteins. We could also amplify samples of DNA by PCR and use it in gene sequencing to predict certain determined conditions. And again, we learnt about this with sickle cell anemia, where there is a mutation, a point mutation, where the mutation in the codon codes for the amino acid valine instead of glutamic acid. So here a thymine substitution mutation substitutes for adenine in the DNA sequence. So effectively, gene sequencing can be used to predict possible links to genetically determined conditions such as sickle cell anemia by looking for mutations. DNA samples can also be amplified by PCR because you could use them in forensic science. An example of this would be in DNA profiling. So in this case, you'd want to extract your DNA and increase the number or the quantity of DNA samples using polymerase chain reaction. So DNA extracted from cells and amplified using PCR. And then if we're using forensic science, you'd look for two different DNA from people and you want to compare them. And you could see here that obviously there would be some differences between people. In this case, person 2 has got a longer DNA fragment because when you cut the DNA using restriction endonuclease enzymes, you obtain short tandem repeat sequences from DNA. You can see in person 2 they've got an extra CG repeat, and the reason they have that's their short tandem repeat is longer, therefore they're going to produce a longer DNA fragment, and that's how you can tell the difference between two different people by looking at their DNA. And some people have got extra or longer DNA fragments because of these additional short tandem repeats, and the length of the DNA fragment is based on how many extra short tandem repeats they have. So therefore you could look at the difference between people in forensic science using DNA profiling. And these DNA fragments can then put into a gel electrophoresis. So use a gel electrophoresis to separate the DNA fragments. And obviously we have the idea that shorter DNA fragments will travel much faster down the gel, whereas the larger DNA fragments will move much slowly. And to visualize these DNA fragments, you can use UV light, you can use radioactive probes, fluorescent probes, or southern blotting. And then what you want to do is you want to compare the different patterns of bands. So you could do that to identify criminals. So here you've got the DNA from a crime scene, which you could amplify the DNA by PCR for forensic science. And this is an example of DNA profiling to identify criminals. And when you compare the DNA from the crime scene, when you're comparing it to suspects, you're looking for the same combinational pattern of DNA fragment bands, because obviously the DNA would be cut using restriction endonuclease, and that will produce the same size DNA fragments. So the suspect will always have the same DNA fragment size, and as long as the combination pattern of DNA fragment bands is the same, then clearly that suspect is your criminal. Another reason you could use in forensic science of DNA profiling is to test for paternity. And here, all the DNA fragment bands of a child is inherited only from the mother or the father. So in the example, you can see here that some of the DNA fragment bands come from the mother, and then some come from the father. Every single DNA fragment band must come from either the mother or the father. They can't come from another person. So that's how you could identify who the mother and the father are. So just to repeat this, you need to use restriction endonuclease to cut the DNA. The length of the DNA fragment clearly can be different due to the short tandem repeats. You can then put them in a gel electrophoresis to separate the DNA fragments. The shorter DNA fragments travel further, the longer DNA fragments travel much more slowly, and this produces a pattern of bands, and those pattern of bands can then be compared. So an example, as we mentioned, was in crime scenes to find out if the suspect is a criminal, or if we're looking at paternity, clearly half of the DNA fragments must come from the mother and the other half must come from the father. They can't come from any other individual. And then the other thing we could do also is to sequence the gene, as we mentioned earlier, to look for predetermined conditions, mutations such as sickle cell anemia, or to then predict the amino acid sequence of proteins. 7.2 Factors affecting gene expression. In topic 1, we learnt about protein synthesis, and in transcription, we know that you start off, or the whole purpose of transcription is to produce mRNA, which is a copy of the DNA genetic code. In other words, it has the same base sequence, and this involves using a helicase enzyme, which unzips the DNA by breaking the hydrogen bonds, and therefore you've separated your DNA sequence of the gene with the opposite strand, which is the template strand, and then you use the template strand to produce the mRNA, and thus requires the use of RNA polymerase. And the mRNA is made by complementary base pairing 3 RNA nucleotides to the template strand, and then these RNA nucleotides adjacent ones have to be joined together. So that means phosphodiester bonds have to form between them, and again that's done by using RNA polymerase to make the mRNA. And that mRNA obviously would then leave the nucleus through the nuclear pore and attach to ribosomes in the cytoplasm or in the RER. Now, to do transcription, that means that the DNA has to be accessible to RNA polymerase. And DNA is actually wrapped around proteins, and these histone proteins can make DNA inaccessible. And modifications of the DNA can either make them inaccessible or make them accessible, depending on which type of modifications there are. So on the picture on the top right, you can see that methyl groups are added, and this makes the DNA inaccessible. But in the lower picture, acetyl group addition causes the DNA to be accessible so that you can do transcription, so that RNA polymerase now does have access to produce the mRNA. And then during this stage, when you're making the mRNA, not the whole of the mRNA is actually used to then code for specific amino acids to put in the correct amino acid sequence. So the RNA that is produced, or the pre-mRNA, actually has exons and introns, and you can then splice them in different combinations. So the same gene, the same DNA sequence can code for different proteins that are very similar to each other, because you could splice different parts of the DNA sequence and combine them in different combinations. And when producing the mRNA, that mRNA can then move to the ribosomes for translation to occur. And then during that translation, it will then code for different proteins, even though you started off with the same DNA sequence from the gene. So this effectively is trying to create the same food, but you're either adding one additional ingredient or using slightly different ingredients. You could be substituting the ingredients or using the ingredients in different combinations. But effectively, you're still making the same food. It's just that there might be a slight variety of the same food. Define transcription factors. Transcription factors are proteins that bind to DNA and affect transcription. Explain role of transcription factors in regulating gene expression. So transcription factors have DNA binding regions that enable them to bind to promoter sequences, and these promoter sequences are located just before transcription starting point of a gene. And they can help to stimulate the start of transcription. So that is the purpose of transcription factors. However, there are other transcription factors that bind to enhancer sequences, and they change the chromatin structure, and this makes it either more open, so in other words, the gene expression could be active, or less open, so the gene expression is inactive, to RNA polymerase, and we saw those examples earlier. Explain how post-transcription modification of mRNA in eukaryotic cells, in other words, RNA splicing, can result in different products or different proteins from a single gene. So as we mentioned earlier, a single gene with its DNA sequence could actually code for different proteins because of the way you splice the RNA to produce the mRNA. So pre-mRNA producing nucleus from transcription includes exons, which are the coding part of the DNA, and introns, which are non-coding DNA. So those introns don't actually code for amino acids, but the exons do. But how much of the exons you want to use, or which combination you want to use, can therefore be used to produce different proteins. So modification of pre-mRNA involves removal of the introns, because they don't code for specific amino acids, and then sometimes some exons, so depending on which exons you do not want to include. So non-removed exons are joined together by spliceosome enzyme complex to produce mRNA that can then be translated. Effectively, that mRNA can then go to the ribosome to be translated. This RNA splicing results in a single gene having possibility to produce different polypeptide chains or proteins, and thus different phenotypes. So in the example that we just saw, you could splice the pre-mRNA, so you have exon 1, 2, 3, and 4, but then you can use different combinations. You can splice it to have exon 1, 2, and 3, or exon 1, 2, and 4, or exon 1, 3, and 4. So therefore, by splicing in different combinations of the exons, you're producing different mRNA from the same DNA sequence, and therefore you end up with different types of the protein, and this could result in people having different phenotypes. So again, using the same idea, you're producing the same protein, such as using the ingredients to make the same food, but you could leave certain ingredients out or add additional ingredients to make the same type of food, but make that food slightly different to each other. So in other words, you're ending up with proteins that are very similar to each other, but they have a slight different functions. But all of those proteins were produced by being coded from the same DNA sequence that was then spliced during the pre-mRNA stage. Describe epigenetic modification. Epigenetic modification is changes that affect gene expression. So we saw some examples earlier. You can see an example here. We've got a cytosine base, that cytosine base is being methylated, and that methylation prevents the expression of the gene. So DNA methylation, non-coding RNA, and, with examples we saw earlier, histone modification or histone methylation or acetylation can be used, and this involved in change to protein synthesize or cell differentiation. So that's the effect that epigenetic modification has. You could either synthesize different proteins, or you could also affect how a cell differentiates. So we start off with DNA methylation. So you can see there's a methyl group, CH3, is added to the CpG site. And a CpG site is where there's a cytosine next to a guanine with a phosphate bond in between. So C cytosine, P is the phosphate bond, and G is the guanine. And that's to silence gene by preventing transcription. So DNA methylation, where the methyl group is added to a CpG site, silences gene by preventing transcription. In terms of histone modification, so these are the proteins that DNA is wrapped around, and if you do histone methylation, that's the addition of methyl group. So this is the methyl group added to the histones, not to the DNA. So that CH3 causes inactivation of DNA, just like DNA methylation, and that silences the gene. Because effectively you're also preventing transcription. You're making the DNA less accessible for RNA polymerase. So both types of methylation, DNA and histone methylation, causes inactivation of the DNA and silences the gene to prevent transcription. However, histone acetylation is the addition of an acetyl group, which is COCH3, and that opens the chromatin structure, which allows gene transcription. So in other words, you're enabling transcription, you're enabling the RNA polymerase to have access to the DNA so that gene transcription can occur. Explain how demethylation of DNA converts embryonic fibroblasts into pluripotent cells. So demethylation is the removal of the methyl groups. So demethylation results in a gene being activated. Because remember, the whole point of methylation, whether it's methylation of the DNA or methylation of the histone, is to silence the gene, is to make the DNA inactive to prevent transcription. So demethylation will be the opposite, means that genes can be activated, and therefore transcription of a gene can take place. And when that transcription takes place, that produces proteins that causes changes in the fibroblast cells. Describe how epigenetic modification is important in. So here we start off with a single cell, and in that single cell, the expression of neuron-specific genes causes the conversion into a neuron. However, using the same genetically identical daughter cells, if there was an expression of epithelial-specific genes, it could then convert into an epithelial cell. So therefore, the cell has all the genes, but depending on which specific genes are switched on or switched off, will have an impact on which sort of function it has in the future. So it's important in ensuring cell differentiation. In other words, making cells different so that because they have different functions. So cell differentiation takes place as unspecialized cells switch different genes on or off. And that's another way of saying the genes being expressed or not being expressed to become specialized cells. Most changes are results of epigenetic modification. So the examples we saw were things like histone acetylation or DNA methylation, transcription factors. All of those are examples of epigenetic modification that causes cell differentiation to take place. In other words, it affects the genes being switched on or off, and those specific genes being switched on or off then cause unspecialized cells to become specialized cells because they ensure that very specific proteins are made within the cells, and those specific proteins therefore will then determine the specific function of the cell, so the cell then differentiates into those different types of cells. An example of this is alpha, beta, and gamma globin genes when they're being switched off or on in erythrocytes, which are red blood cells. We know that in erythrocytes we've got hemoglobin, 2 alpha and 2 beta in our bodies. However, when you start off as a very early fetus, you actually have alpha and gamma globin genes switched on, and the beta globin genes are actually switched off. And this increases up to 6-week-old fetus, where you've got a very high percentage, or 50% of your globin genes are alpha. And as you keep getting older, it gets to the state where just before birth, the expression of the globin gene switches. So in other words, you've got gamma globin genes that get switched off. They're not expressed anymore, but then the beta globin genes get switched on, and therefore you're starting to produce more of the beta globin genes. So you're switching from when we learnt about fetal hemoglobin to now producing adult hemoglobin. And that clearly happens in the same cell because you've got those same genes, but you're expressing different specific genes by switching them on or off. And that clearly results in different phenotypes. In topic 2, we learnt about how early development of embryo to the blastocyst stage happens. And we know that when we start off, we've got your zygote, which is the fertilized egg, but then also going from the zygote to the morula, which is very early embryo, those stem cells are totipotent, which means that they have the potential to differentiate or specialize into all cells. Now, from going from the zygote to a very early embryo stage, we've got a cleavage process where there's an increase in cell numbers by mitosis. However, this happens without growth. In other words, you have a very shortened interphase stage. So those cells become progressively smaller, and they form a hollow embryo structure, a blastocyst, with a thin cell layer surrounding the fluid-filled cavity. Now that blastocyst, which is a late embryo, contains the inner cell mass and the trophoblasts, or trophoblastic cells, which is the outside part of the blastocyst. And the inner cell mass contains pluripotent stem cells, and they give rise to embryonic cells, or most cell types, but not the extra embryonic tissue, such as the placenta. They can only differentiate to limited cell types, so they've stopped being totipotent stem cells, they're now pluripotent stem cells. Whereas the trophoblastic cells, which is the outside part of the blastocyst, they become the extra embryonic tissue, such as the placenta. So they are no longer totipotent and pluripotent as well. Explain what is meant by stem cells. So if you use the definition from the previous slide, we understand that stem cells must be undifferentiated cells that have the potential to develop into many different types of specialized cells. And then we've got to look at the differences in the potency. So obviously we know that they could be totipotent stem cells, or pluripotent stem cells, or multipotent stem cells. The totipotent stem cells, as we mentioned earlier, can be found in the zygote, which is the fertilized egg, or in the morula, which is very early embryo stage. The pluripotent stem cells are in the blastocyst, which is a more later embryo, and then the part of the blastocyst that contains the pluripotent stem cells are the inner cell mass. Whereas multipotent stem cells are in the developing embryo. So after the blastocyst, that embryo then continues to develop. So that developing embryo will contain multipotent stem cells, and we still have those multipotent stem cells in our body. They're known as tissue-specific adult or somatic stem cells. And they become only some cell types. So they're already partially specialized. They can't become every type of cell. They can only become certain types of cell. Explain why stem cells from fetal cell lines are being used to produce Ebola vaccines rather than bone marrow cells. So clearly bone marrow cells would be the tissue-specific adult or somatic stem cells, whereas the fetal cells would contain pluripotent stem cells. So why would it be better to use pluripotent stem cells than multipotent stem cells? So fetal stem cells will be pluripotent, therefore they're able to develop into a wider range of cell types than the multipotent or the bone marrow cells. Explain why some cells are not able to become other cell types. So obviously, if you already decide to specialize, that means that you've got expression of certain genes that are turned on, but then other genes are turned off, and you can't reverse that. So using that idea, because they are fully differentiated or specialized cells, they're not able to express certain genes. So that means genes needed for other cell types are permanently switched off or silenced. Therefore, proteins needed by other cell types can't be produced. And obviously those proteins will determine what sort of function your cell has. So if you can't produce those proteins because the genes required to produce those proteins are switched off or silenced, you can't then become other cell types. In other words, you've already become fully differentiated or specialized type of cells. And here we can see an example where we go from totipotent stem cells to pluripotent stem cells. So we start to differentiate and then become multipotent stem cells, such as the one found in our tissue-specific adult stroke somatic body cells. And then eventually, once they fully differentiate and become specialized, they become unipotent. In other words, they've lost the ability to become a different type of cell. So as the stem cells differentiate, the potency decreases, the capacity to differentiate into specialized cell types start to decrease. Now we can use these ideas because if we look at tissue-specific somatic or adult stem cells, they are found in specific tissues such as the skin or muscles, umbilical cord, brain, heart, bone marrow, and we can utilize this. So we know that they are partially specialized. In other words, they've already started to become specialized, but they're not unipotent yet. And using that idea, we know that they will then start to differentiate in the future. And this occurs through mitosis, where these tissue-specific stem cells first divide by mitosis, and then they differentiate to become specialized. And this can be used in medicine. So for instance, explain how stem cells could be used to help repair damaged heart muscle cells in someone who has a heart attack. So what we could do is we could get some stem cells and inject it into the person's heart, and they would then differentiate into those new heart muscle cells. And what you're doing is you're not repairing the cells that are damaged, you are replacing it with the stem cells that have been injected, which then differentiate into the new heart muscle cells. And effectively, you're then helping by replacing those damaged cells, you're helping to repair the tissue or the organ, which in this case is the heart. However, there are problems with using stem cells, and this you should have learnt in GCSE. Obviously, as mentioned before, problem is that these stem cells do mitosis, and if they do mitosis uncontrollably, then one issue would be the fact that they could then develop into cancer. But then the other issue is the fact that the person's immune system might attack the stem cells, and therefore those stem cells could get rejected. What are the ethical issues with using embryonic stem cells in stem cell therapy? The problem with using embryonic stem cells is clearly they have the potential, those destroyed embryo, to become living human. So when you're using embryonic stem cells in stem cell therapy, the embryo gets destroyed, and effectively this means that you could be destroying something that turns into a living human. So one way to get around that is by using induced pluripotent stem cells. So this is where you take cells from a body and you genetically reprogram it to become pluripotent stem cells. So we've already got unipotent cells in our body. In other words, we've got somatic cells in our body, but if we could genetically reprogram it and turn it back into a pluripotent stem cell, we can then insert that pluripotent stem cell. Well, first we'd have to make more of them in a lab, and then by using various factors we could then differentiate into the cell types. And clearly this is going to be ethically better because we're starting off with cells already in our body. We're not destroying an embryo. So now we need to know, how do you make these induced pluripotent stem cells? So you can see an example here. We start off with adult fibroblast cells. We could reprogram the cells using the proteins or certain genes that express those proteins to produce the iPS cells, and then those iPS cells can be grown, and after growing them you could then differentiate them. Explain how to form induced pluripotent stem cells or iPS cells. So you'd start off with your differentiated fibroblasts, which is non-pluripotent. In other words, they come from your somatic cells from a person. And then you'd want to artificially introduce genes into the cells. So these are reprogramming genes for transcription factors that will clearly then affect how these cells are specialized. The examples you want to use in exams are OCT4 and SOX2. These are the 4 common ones, but memorizing the first 2 would be helpful for the exam. And when you artificially introduce them, you'd want to use a virus vector, and then these iPS cells can be cultured in growth hormones, so you can make more of them. And once you've done that, you could then transplant them, in other words, inject them into the person's damaged tissue or organ. So effectively you're replacing those damaged cells, not repairing them. Describe how induced pluripotent stem cells could be used to cure retinitis pigmentosa. So this clearly has to do with the eye. In this case, you'd replace, again we're using that word, dysfunctional cells with functional cells derived from iPS cells. The iPS cells are injected into the retina and they will then develop into the normal retinal cells. Explain why use of iPS stem cells may be less problematic than embryonic stem cells, including compare and contrasting their properties. For induced pluripotent stem cells and pluripotent embryonic stem cells, they both have the potential to divide indefinitely because they're both pluripotent. They both have the potential to differentiate and or specialize into a number of cell types. The iPS cells were adult cells. They were cells taken from, or somatic cells taken from the body, whereas the pluripotent embryonic stem cells were taken from the morula or the inner cell mass of the blastocyst, and that's why they are pluripotent. No gene is added for these embryonic stem cells. However, you have to add genes such as OCT4 and SOX2. So these are the reprogrammed genes for transcription factors. Therefore, there is no or less ethical issues with the iPS cells. However, the embryonic stem cells have ethical issues because a destroyed embryo therefore had the potential to become a living human. The iPS cells can form adult cells, whereas the embryonic stem cells form younger cells. However, they could be antigenic, so you could have issues with rejection. However, because the iPS cells were made from somatic cells from the same person, they will produce patient-matched cells, so there's no rejection issues when using induced pluripotent stem cells. 7.3 Gene Technology. In GCSE, you would have learnt about genetic modification, and obviously there are lots of stories in the last couple of decades about them. So genetically modified crops can save us from food shortages. Genetically modified pigs are best bet for organ transplant. Demand for designer babies to grow dramatically. So obviously there are some headlines that are very pro and some headlines that could be against, but they're very, very dramatic headlines. So first of all, what is genetic engineering? And here's an example you'd looked at before, venomous cabbage. So the scorpion is venomous, but the cabbage is not. So how can you make the cabbage venomous? Well, if you look at these keywords, what we could do is genetic engineering is when you artificially change the DNA of an organism, in this case the cabbage, we're changing its genome by inserting genes from another. So we're inserting the venom gene from the scorpion into the cabbage, and therefore we could produce organisms, a venomous cabbage, with the desired characteristic. In other words, making the cabbage venomous. So using the idea of protein synthesis, if you've got the instructions, the genes, you can therefore make the proteins from that instruction. But we can take that instruction from an organism and put it into a different species so that the transgenic organism, the new species, or the species that we're going to insert the gene into, now has that instruction and can also produce that protein. And clearly, we're introducing genes artificially. These are genes that they would not naturally have. So why would you want to do this? In this case, the venomous cabbage, we start off with the scorpion, and the gene that we're inserting into the cabbage is the venom gene. And the big reason for doing this is the venom is poisonous to caterpillars, so it can act as a pesticide. Clearly, you wouldn't use a venom that's poisonous to humans. It'd just be one that's poisonous to caterpillars, and that would be very helpful. And note that the cabbage wouldn't naturally have this venom, but now it has the instructions to make it because we have artificially inserted the gene which we obtained from the scorpion. Other examples you would have learned in GCSE are things like golden rice, where you could take the vitamin A gene from carrots and put it or insert it into rice, and that helps with vitamin A deficiency. Or spider goat is a good example where you've got spider silk. The spider silk gene is inserted from a spider to a goat, and the goat then produces it in its milk, and therefore you could produce a larger quantity of the spider silk because the spider silk gene or instructions to make the spider silk protein is now in the goat, and you could use it for lots of examples such as bulletproof clothing. So how is this done? Well, first you're going to have to isolate a gene from an organism that already has it. And in this case, to isolate it, you'd use a restriction endonuclease. So we learned previously that you cut DNA using restriction endonuclease, and when you do that, you produce the sticky ends of unpaired bases. Then when you're going to insert this gene, to insert it, you're going to need something that sticks DNA together. In this case, you'd use a ligase enzyme. So the ligase job is to stick those sticky ends together. And here's an example. We've got our plasmid, which you could take from a bacteria, and we use a restriction enzyme. You can see it produces the sticky ends, and then we could isolate our desired gene, and if we isolate it, it will also produce sticky ends if we use the same restriction enzyme, and those sticky ends can then join together using ligase enzyme. So here's the same example. We've isolated our gene that we're interested in. We use restriction endonuclease that produces the sticky ends. We use the same restriction endonuclease in a plasmid vector from a bacteria, producing the same sticky ends. We could then stick or join these sticky ends together using DNA ligase. In this case, we're using a plasmid to transfer the gene. So that plasmid is a vector. So the plasmid vector we got from a bacteria can then infect an organism, and the gene can therefore be inserted into the organism. So plasmid is a type of vector that can be used, and obviously we're using bacteria in this case because that's where they're naturally found. But other types of vectors you could use to transfer the DNA or the gene can be a virus. So you could put it into a virus and the virus can infect and transfer the gene. There are other types of vectors as well, which we'll mention. So genetic engineering involves inserting a gene from one organism into genetic material of another to modify its genome. In other words, you're changing what's in the DNA. Explain how recombinant DNA can be produced. Since you learnt this in GCSE, you've got your isolated desired gene, and that's using restriction endonuclease. However, there is another method where you can take, alternatively, the transcribed mRNA. So that desired gene will produce mRNA during transcription, and if you get that mRNA, the problem is that it's a single strand, so you could use an enzyme called reverse transcriptase to convert it into a double strand, but this double strand, because you started off with mRNA, we would call it a complementary DNA or cDNA, but it would still have the same order of bases or DNA sequence as the isolated desired gene that you got using restriction endonuclease. Then you'd want to insert the desired gene, or you could insert the cDNA. Remember, you've got sticky ends, and you'd insert it into the plasmid vector, and because you use the same restriction endonuclease to cut the DNA, the desired gene in the plasmid vector would have complementary sticky ends, so those sticky ends can be joined together using DNA ligase due to the complementary base pairing. So effectively hydrogen bonds will form between them. Explain how recombinant DNA can be inserted into other cells. So we already know that we'd have to use a vector, and we've mentioned two different types of vectors already. So vectors transfer desired gene into new cells to incorporate into the host genetic material. So we've got plasmid vector, which we get from bacteria. We've got virus vector, which can infect a host cell by inserting the desired gene. We could also use a gene gun vector. This is when DNA is shot into a cell at very high speed, and if it's successful, that DNA then is now part of the genome of that cell. Explain process for genetic modification of soybeans to improve production. So here, if we look at this example, you'd have to use a restriction endonuclease to cut the gene or plasmid. Therefore, you produce the same sticky ends. You'd use ligase to insert the gene. Then you'd have to use a plasmid vector to transfer the gene. When you've got your plasmid vector, you'd have to put it into a bacteria to help transfer of the recombinant plasmid. And in this case, the bacteria you use is Agrobacterium, and then that could transfer the recombinant plasmid directly into the plant. And when it does that, the infected cells develop into a growth tumor, which is a crown-cord tumor, and we would then use cloning of modified transgenic cells to produce a whole new transgenic plant. Enzyme desaturase converts oleic acid into linoleic acid. Soybeans can be genetically modified to produce less linoleic acid. Explain how these genetically modified soybeans can be produced. So in this case, we want more oleic acid instead of linoleic acid. So we want to get rid of the enzyme desaturase. So you can remove or replace desaturase gene, or use silence or knock out the gene, or induce mutation on the gene, therefore it's not functional, or reduce the activity of the promoter, so of the gene. And then using restriction endonuclease to cut the gene in the plasmid, produce the same sticky ends, and then you'd use ligase enzyme to stick those sticky ends together to insert the gene into the plasmid. Use a plasmid vector to transfer the gene. Use Agrobacteria to transfer the recombinant plasmid directly into plant. And then you could transform the cells grown into the adult plants via tissue culture. In other words, you're going to clone so that you make sure that these transformed cells grow into lots and lots of adult plants. Explain how to identify recombinant cells. So you've got your bacterial cells, and you want to make sure that the bacterial cells has taken in the plasmid, but then you also want to make sure that the plasmid has the inserted gene as well. So how do we make sure that we've got recombinant bacterial cells that not only contains the plasmid, but the plasmid also contains the inserted gene? And we could do this because cells with recombinant DNA can be identified if the recombinant DNA inserted into the plasmid carries marker genes for antibiotic resistant. So in other words, we have to look at the plasmid itself, and the plasmid has got two genes that are resistant to two different antibiotics. So in other words, you've got a gene that is resistant to antibiotic 1 and a gene that is resistant to antibiotic 2. So you can see that in the diagram, the top plasmid, it's got two different types of genes that are resistant to the two different types of antibiotics. And therefore, our results could be three different things. We could either have a bacterial cell or a recombinant bacterial cell with the recombinant plasmid inserted, therefore that plasmid contains the inserted gene. We could have a bacterial cell that does have the plasmid, but the plasmid did not contain inserted gene, or we could have a bacteria where there's no plasmid inserted, so it doesn't matter if the gene is inserted in the plasmid, it just hasn't been taken into the bacteria. And clearly those are the three options, and depending on how you treat the bacteria with antibiotic 1 or antibiotic 2, we can then figure out or identify if the bacteria is a recombinant cell. And this depending on the results of how we treat those bacteria. So to identify the first option, the one that we want, if we were to put recombinant plasmid, the one that contains the inserted gene and contained in the plasmid, and that plasmid is in the bacteria, therefore if we treat it with antibiotic 2, the antibiotic 2 will be in the plasmid. So if the plasmid has been taken up by the bacteria, that means that the bacteria will survive because it contains the plasmid which contains the resistant gene for antibiotic 2. So as long as the plasmid has been taken in by the bacterial cell, it will survive when we treat it with antibiotic 2 because the plasmid contains the resistant gene to antibiotic 2. So therefore, when we look at 1 and 2, both of them have plasmids inside it. Therefore, when treating with antibiotic 2, that bacteria will survive, and that's how we know that the bacteria has got plasmid inside it. If the bacteria don't grow, that means there was no plasmid, and because there's no plasmid, therefore there's no marker gene 2, there's no resistance to antibiotic 2, so therefore the bacteria will not grow, they will die. And so we could differentiate with the third type of bacteria that contains no plasmid inserted. So how do we now know if the plasmid does contain the inserted gene? Well, this got to do with the gene for resistance to antibiotic 1 being disrupted. So if the inserted gene goes into the plasmid, what it does is it cuts in the middle of the resistant gene. Therefore, that resistant gene to antibiotic 1 is non-functional. So therefore, that plasmid will not be resistant to antibiotic 1. So an inserted gene will disrupt the restriction to the resistant to antibiotic 1. Therefore, that bacteria will not be resistant to antibiotic 1 anymore. So when you treat it with antibiotic 1, those bacteria with the desired gene inserted into the marker gene 1 will no longer be resistant to antibiotic 1. They will die. However, if the inserted gene, or if the gene that we desire is not inserted into the plasmid, it will still have resistance to antibiotic 1, so it will survive. So we want a system where treating it with antibiotic 2 means that the bacteria survives because it has the plasmid, but when you treat it with antibiotic 1, it dies because the insertion of the desired gene disrupts the gene that produces a resistance to antibiotic 1, and therefore there's no more resistance to antibiotic 1. So genes of interest is inserted into plasmids. The plasmid has a marker gene, so obviously this marker gene is for antibiotic resistance. The recombinant bacteria will be resistant to this antibiotic. The bacteria is grown on agar containing the antibiotic, so we're talking about antibiotic 2 in this case. Only recombinant bacteria will grow. However, then you could use something called replica plating, which we'll explain later on, to transfer the recombinant bacteria to fresh medium. And obviously, if you then test it out on antibiotic 1, those bacterial colonies that die because the resistance to antibiotic 1 gene has been disrupted, they must therefore contain the inserted gene. So by replica plating, you could then identify which bacterial colonies or cells contain the plasmids with the inserted desired gene. The whole point of replica plating is it keeps a living copy of patterns of bacterial colonies. So in other words, when you've got your bacterial colonies, you do replica plating, and that imprints all the colonies, so you could then test out the imprinted version on antibiotic 1, and if they die, you will spot that there are no bacterial colonies in that location, but because you've imprinted them from the original, because you produce this replica plating, you could then identify which bacterial colonies have your inserted desired gene in the plasmid, and the plasmids are also in the bacteria. So what is the whole purpose or advantage of doing genetic modification? In other words, characteristics we might want in crops are clearly going to do with resistance to things like pests or environmental conditions such as frost, disease, being resistant to herbicide, drought, or having a longer shelf life, or possibly increasing the yield. However, there's disadvantage because those genetically modified crops could cross-pollinate with wild plants or weeds, and therefore the offspring of those weeds will therefore have the genetic modification, the genes, and therefore we can't use them anymore if we're using herbicide. Explain how knockout mice can be used as a valuable animal model to investigate gene function. So knockout mice has got to do with making a gene non-functional and then seeing the response in the phenotype or the change in the phenotype. So genes knocked out to produce animal models of disease, as well as helping to understand function of genes, mice can be produced with genetic disorders similar to humans. These mice can then be used to test possible treatments and investigate problems. So an example would be mice developed with faulty CFTR gene, which causes cystic fibrosis in humans. New gene therapy treatments can then be tried to demonstrate safety before human trials, and this speeds up development of potential new treatments. So if we know that mice contains genes that are similar to us that we suspect could cause genetic diseases, we could then knock them out on the mice and then use treatment on that mice to see what the effects are. And obviously, therefore, you could do this before you do human trials, and that would give you the advantage because you'd be able to create mice with similar phenotypes to the genetic disorders and see what sort of treatments work in the mice. Explain why widespread use of genetic modification of major commercial crops and other transgenic processes cause public debate of their advantage and disadvantages. So in terms of concerns, the marker genes may get into wild plant population, building up antibiotic resistance. Ingesting foreign DNA, although these genes generally exist already in food. So even if you take it from one organ to another, you might already be ingesting the proteins that are produced anyway. There's a strong objection to animal use in scientific research as they violate animal rights, and making new GM strains that tend to be infertile, meaning new seed brought early. So one of the problems is that sometimes when you make these seeds from genetic modification, they tend to be infertile, so you have to continuously buy new seeds. And that is the whole of topic 7, modern genetics.